Yeah, so what we kind of already gone over here was this on the top right over here is the, a section of a myofiber, right? A muscle cell, right? You could tell by the striations. Here's the nucleus of the cell, and that muscle cell was packed with the myofibrils, the contractile organelles, right? That were a series of those sarcomeres, right? Stacked end to end. Then as the muscle actually contracts, right? What happens? That muscle actually gets shorter, right? And it's getting shorter when the muscle fiber itself contracts. What happens? A whole bunch of them contract enough and the whole muscle contracts, right? Your whole muscle gets shorter and creates a pulling force, right? This is just one muscle cell doing it. And we'll see, as we saw, it was because of these myofibrils that were actually contracting, right? And if we look a little closer at all this, right, the myofibril down the whole length of the muscle fiber is contracting because these series of sarcomeres, right, from these Z discs in red here, so here's sarcomere 1, 2, 3, 4, right, all along the length, right? They were packed in, arranged. These are individual myofibrils packed inside the muscle. And such it was, you had the red filaments here, the thin filaments in red, the actin filaments, and then the myosin filaments. And they're arranged in such a way that they would slide over each other and down the length of the muscle, right? So when they did that, the whole myofibril contracts, and they're all going to contract at once within a muscle cell, right? So the whole muscle cell is going to get shorter and then create that pulling force. So that's what we kind of went over last time, and all the elements, the Z disc, right? The I band, the A band, the Z disc, the M line, H zone, M line, right? All those things and how they changed as we moved along there. Shorten, and shorten like that, right? You can see how everything changes. They just slide up against each other, right? No proteins get shorter with the exception of titin, right, which isn't really involved in the sliding filament per se, although it does have important functions. But the actin and myosin filament, they just change in relationship to each other like a sliding ladder, like a expandable ladder, right? So today then we're going to be talking a little bit about more of the physiology, right? The interaction between myosin and actin, right? How they actually interact with, and what the role of ATP in this sliding filament theory, and the different role of calcium, right? So we're going to look at the ATP and calcium for the most part. And then we'll look at the concept of excitability and how an action potential, right? We're going to introduce this concept without going too deep into it because we're going to cover this in the nervous system, and then see how this excitation of the muscle fiber is related to the contraction that we just saw right there, right? So here is the, all the structures we went over, right? The I band, right, which had the Z line, the Z discs in the middle, right? So from Z line, I'm sorry, I know I keep Z line or Z disc means the same thing. From Z line to Z line is the sarcomere, and off the Z line were the actin filaments on each side right there. And they had some overlap with the myosin filaments, right, which were kind of the stable part in the middle attached to the M line over here. And during contraction, those actin filaments were pulled in toward the M line over here, creating a greater overlap with the myosin, and that's what caused the contraction, right? And so what we're looking at today is, like, you see these as kind of straight lines, right? Just some kind of abstract, but you see these little nubs, right over here, right? That's kind of the interaction is what we're going to get into today. So again, over here, and just to keep the big picture in mind, right? We have these sarcomeres that are going to be end to end within the myofibrils, which are packed inside the myofibers, which make up the muscle, right? They were in the fascicles, which make up the muscle. So that when this contraction happens, that's why your muscle contracts. It creates this pulling force on the bones through the tendons. Right there, right? Thousands and thousands of these within here, right, is going to match up with what's happening in your muscle eventually, that you can see and feel on your own body right there. That's what's happening deep inside right there. We want to look at these little tiny little nubs right off here coming off the myosin elements right there, because these are going to be the important interaction points between myosin over here in brown and the actin filaments over here, right? And then so, looking over here, right, here's that myofibril section, these two Z lines or Z discs. See, they keep changing the names here. Z discs right here, over here, make up the sarcomere. Here is a half a sarcomere. Right here, here's the M line. Here's that half of the myosin filament. Here's the Z discs, and here is the actin filaments. And you see coming off the M line over here, you have that part of the myosin, right, which starts to give rise to these myosin heads over here, right? These are these structures over here, right? And then these are the myosin tails, right? So you can see in the midsection around here, right, it's all the myosin tails, and then as you move out laterally, you see all these myosin heads sticking out, right? And this is what's going to be interacting actually with the actin filaments, right? So notice over here on these myosin heads, right? This is the structural kind of part of it that's going to interact, but there's also the interaction point with the actin filament, as well as this ATP binding site, right? This is going to have this enzymatic activity on this part of the myosin head right here. So remember remember ATPases, what they do, right? Way back when we talked from the first unit, we talked about these enzymes, right? That hydrolyzed ATP to ADP, right? Which is going to cause this conformational change in there, right? It's gonna create energy by hydrolyzing the ATP. You're going to remove a phosphate group from the ATP to form ADP, a triphosphate to a adenine diphosphate, right? Plus an inorganic phosphate. So this removal of the phosphate group, that hydrolyzation, right? When you add a drop of water and the enzyme kind of catalyzes all that, it creates energy, right? It creates stored energy. So the energy that's created by this ATP hydrolysis is going to be trapped in the myosin head right here, and it's going to be stored as potential energy. And so at some point along the line, your myosin head is going to bind ATP and hydrolyze it, and it's going to be energized. All right. And when it's energized, it's going to said to be cocked or ready to rotate. That is, if this was a gun, right, this is the pulling of the trigger, like cocking of the trigger right there, like getting it ready to fire that ATP hydrolysis, right? So this potential energy that's formed by this binding of ATP over there is going to convert it into kinetic energy, into the power stroke that's actually going to move the actin filament toward the M line. All right, so what we're going to do is look at the kind of process right here as that whole step happens. And starting from this point where the myosin head with that ATPase activity hydrolyzes the ATP, it becomes energized, right? And it becomes oriented, ready to interact with the myosin, with the actin filament, right? Then, for reasons to be explained later, the myosin head is going to bind strongly to actin and form the cross bridge. The cross bridge formation right over here. Right, so this will come up a lot, right? This cross bridge formation, and later we'll see how exactly this happens. Right now we're just concentrating on the ATP and not the calcium part of it. So after the binding, after this cross bridge formation, you go from what is a 90-degree angle over here in relation to the actin filament, and to a 45 degree. That is, the myosin head is going to pivot, right? It's going to remain stable. It's going to pull the actin filament toward the M line, toward the center of the sarcomere, right? That's called a power stroke. You go from this position here, it pivots and pushes it toward there. Okay, so just to be clear what's happening, right? I think I did this the other day, right? This over here, my hand, my right hand, over here is the myosin, right? And here's the myosin head, my wrist, right? And when it binds to the actin filament, right? It binds, it's at a 90-degree angle, and then it shifts over like that, right? So here's the Z disc, right? Here's the M line, right? It's going to pull it over toward that when it shifts over like that, right? As soon as it bounds, right? The energy to do this over here was from the ATP hydrolysis. And once it binds, it's going to do this, right? That's where the stored energy is being used in this pivot motion. So that's what happens during this power stroke, right? Cross bridge formation, power stroke happens. Actin filament and the Z disc along with it, right? Remember, this is attached right here, is going to be pulled along with it toward the M line right there. And that's what's making the contraction. The last step of all this, so that I can do it again, because it's only going to pull it a little bit away, is that the myosin head again binds ATP, and that allows the cross bridge attaches from the actin, right? So there's no more cross bridge, and you need that step, right? If you don't have ATP, then the myosin head remains stuck to it. That's an important step, right? ATP, the myosin head becomes energized, attaches to the actin, and pivots over. And the other thing to notice is that the ADP and the phosphate group is released during the pivot move at some point, right? So that there's ATP binding ability, right? So the ATP spot is freed up. So here's another view of that, right? Just looking at a different view. We're going to go in a clockwise motion from the sequence of events. This is kind of the inferior side of that, right? On the other side of it. And we're starting from the point where it just went through the power stroke. Here's the myosin head. It has released the ADP and phosphate group and is ready to bind an ATP group, right? So it's stuck. It's still the cross bridge formation here. ATP binds, right? And it detaches. The myosin head detaches, right? And so now it's got an ATP here in yellow, and then it goes through that ATP ACE activity, right? And you go through this ATP hydrolysis. Now you're in the cocked position. After it's in the cocked position, it's in the position to interact with the actin filament. And over here, step number 5, right over here. Here are the power strokes in yellow movement right over here. During the start of the movement here, the phosphate group is released. During the power stroke, the ADP molecule is also released, which frees up the ATP binding site, so the whole cycle can start over again. Okay, so here we are looking, that was a close-up of a single myosin head interacting with the actin filament like this, one little myosin head right here, right? But of course, that's happening down the entire length of the myosin, right? Down toward the Z disc, right? They're all grabbing, they're all pulling again, just like those rowers, right? They're all kind of stroking. And then as soon as that stroke is done, they go and get another one and can pull it further and further, right? Until they can't pull it anymore. So that's happening all down the whole length of the myosin head until it's fully contracted. So that's what's happening, in the role of ATP and all that, right? It gives it its energy, and binding of ATP itself is what allows it to detach from the actin filament. Right? So the hydrolyzation gives it the potential energy to be converted into the kinetic energy of the power stroke, and then the binding of ATP is going to release that myosin head. All right. So here we are again, looking at that. Right? ATP is bound, right? Hydrolysis puts it in the cock position. Power stroke happens, right? And then there's binding, right? But I'm sorry, C happened, the interaction happens, and then the power stroke. But here's where we get into the second part, right? That's ATP. That's the role of ATP in this. Now we want to look at the role of calcium in all this. And what we're going to see is that this interaction between the myosin head and the actin is calcium dependent. And so we'll look at that interaction in the next video. See you then.